Subscribe to Praveen Bhosle YouTube channel and press the bell icon for the latest video updates.
Hello students, let today revise about glycolysis. Glycolysis is also called as EMP pathway because it was discovered by Abedin, Meyerhoff and Parnas. Glycolysis is the first major process in the mechanism of aerobic respiration. It is an anaerobic process since it is independent of oxygen. It takes place in cytoplasm, hence also called cytoplasmic respiration. It is a common pathway for aerobic as well as anaerobic respiration. Glycolysis is defined as the stepwise enzymatic breakdown of the hexose sugar glucose into two molecules of pyruvate in cytoplasm. Glycolysis consists of two major phases: preparatory phase and cleavage, oxidative and payoff phase. First is preparatory phase and cleavage. Phosphorylation. Glucose get phosphorylated to glucose 6-phosphate. Here ATP is used. Inorganic phosphate get attached to 6 carbon atom of glucose and forms glucose 6-phosphate. The enzyme here is hexokinase. Isomerization. Glucose 6-phosphate get isomerized to fructose 6-phosphate. The enzyme used here is phosphoglucoisomerase. Phosphorylation. Fructose 6-phosphate get phosphorylated to fructose 1,6 diphosphate. Here ATP is used. Inorganic phosphate get attached to carbon atom 1 of fructose 6-phosphate and it forms fructose 1,6 diphosphate. Cleavage. Fructose 1,6 diphosphate split up into two molecules of triose phosphate. These are dihydroxyacetone phosphate 3 carbon and 3-phosphoglyceraldehyde. Again it is a 3 carbon compound. Isomerization. 3-phosphoglyceraldehyde and dihydroxyacetone phosphate are isomers of each other and are interconvertible. So here DHAP is converted into 3-phosphoglyceraldehyde. Enzyme here is phosphotriose isomerase. So now two molecules of 3-phosphoglyceraldehyde are formed. Oxidative and payoff phase. Oxidation and phosphorylation. In presence of enzyme phosphoglyceraldehyde dehydrogenase, 3-phosphoglyceraldehyde undergo dehydrogenation, that is oxidation, removal of hydrogen. Simultaneously it undergoes phosphorylation, that is inorganic phosphate get incorporated to give 1,3-diphosphoglyceric acid. During this reaction the coenzyme NAD takes up 2 protons and get reduced to NADH2. ATP generation 1,3-diphosphoglyceric acid is converted into 3-phosphoglyceric acid by removal of phosphate group. This phosphate combines with ADP to produce ATP molecule. Enzyme here is phosphoglycerokinase. Isomerization. Enzyme phosphoglyceromutase brings about conversion of 3-PGA into its isomer 2-PGA. In this reaction, phosphate group is shifted from the third carbon atom to the second carbon atom. Dehydration, 2-PGA undergoes dehydration and phosphoenol pyruvic acid or PEPA is formed. The enzyme here is enolase, which catalyzes the reaction in presence of Mg as cofactor. Water molecule is released. ATP generation. On hydrolysis, phosphoenol pyruvic acid loses a phosphate group, that is dephosphorylation, and gives pyruvic acid or pyruvate. The released phosphate group combines with ADP and ATP generation takes place in presence of the enzyme pyruvate kinase. One glucose molecule is converted into 2 molecules of pyruvate at the end of glycolysis. As each reaction is the oxidative and payoff phase occurs twice, there are 2 PGAL molecules. Therefore, 2 ATP molecules are generated at step number 7 and 2 more ATP molecules at step number 10. In all, it produces 4 molecules of ATP. 2 molecules of NADH2 are produced at step number 6.